And the third reversible inhibitor types is the uncompetitive inhibitor, and this is something different from the competitive and non-competitive, where the uncompetitive inhibitor binds only to the enzyme-substrate complex. So the enzyme is there, substrate is there, enzyme binds with substrate, enzyme-substrate complex is also formed, and when forming the enzyme-substrate complex, the inhibitor will come and bind and not to the free enzyme. And it mainly refers the formation of ES complex. And this uncompetitive inhibition, there are mainly two binding sites. One is for binding substrate and one is for binding the inhibitor, uncompetitive inhibitor. And this complex does not form the product, hence the conformational change of the enzyme. And the example is oxalate, and this oxalate is the uncompetitive reversible inhibitor that mainly inhibits the lactate dehydrogenase. So lactate dehydrogenase enzyme is mainly involved in the conversion of pyruvate into lactate. So pyruvate converted into lactate with the help of lactate dehydrogenase enzyme. And this oxalate mainly concentrates the lactate dehydrogenase enzyme, and it is the product of plant metabolism, and it can inhibit enzymes of lactate dehydrogenase, which interconversion of pyruvate and NADH to lactate and NADH+. So it is a potent inhibitor and can increase even the toxicity of kidney stones, because our excess calcium will get released into the kidney in the form of urine. If suppose there is unabsorbed calcium is there, the calcium will bind in the human body, and this calcium mainly binds to the oxalate. So when calcium and oxalate bind in the human body, it results in the formation of kidney stone and it increases the toxicity. And you could see the example. So enzyme and the enzyme substrate complex is also forms, and after forming the ES complex, the inhibitor will bind, and the active ES complex, when binds to the uncompetitive inhibitor, it creates the inactive enzyme substrate complex, hence no product will be released there, and this is the uncompetitive inhibitor. And coming to the irreversible inhibition, as I told already, it is the permanent loss of enzyme activity, and this is the one does not reverse back once it gets fit into the enzyme, and this enzyme bond is very strong, so the inhibition cannot be reversed. And the very good example is diisopropyl fluorophosphate, DIPF, and it is a nerve gas, so it is mainly affect our nervous system, where it affects our neurotransmitter and it just affects our neuromuscular junction, and it leads to the nervous system damage. So it is an example of irreversible inhibitor, binds to an enzyme acetylcholinesterase. So this acetylcholinesterase is a enzyme in the neurotransmitter acetylcholine. This acetylcholinesterase breaks down to form the acetylcholine, which is mainly involved in the nerve action. So if any nerve action or nerve signaling has to be passed in the body, this acetylcholine will get breakdown with the help of this acetylcholinesterase. When this nerve gas of diisopropyl fluorophosphate enters into our nervous system, it just blocks the activity of acetylcholinesterase, where it damages the nerve muscles, and it also affects our central nervous system, and it also leads to the coma and death condition. So if a patient is affected by this nerve gas, his or her nervous system is completely repaired, and the person finally leads to the coma stage, and some of the paralysis condition also caused by this irreversible inhibition. So once DIPF is attached to the enzyme acetylcholinesterase, it cannot be reversed, and it leads to the permanent loss, and no other paralysis patient get cured completely because of the irreversible inhibition. So this is the overall enzyme inhibition and its types. We saw about enzyme inhibition, what is enzyme, what is inhibitor, and what are the types of inhibition, reversible and irreversible, and in irreversible, competitive, non-competitive and uncompetitive, both very simple mechanism, and in irreversible we have seen a common example of DIBF, and this is all about the enzyme inhibition. Thank you.